So cause and effect to empower drug discovery in a generative AI world. So I might be taking a slightly different stance to some of the talks today. But yeah, let's see how this goes. So first of all, I want to introduce the challenge for drug discovery companies. And this is a slide that nearly every drug discovery company will put up at the beginning of their talk. And it's about the cost of getting drugs to market. So it can be as much as, I think, $6 billion per new molecular entity now. And there are lots of different measures out there that come to these different huge numbers that either include failure rates or not, and lots of different ways of calculating it. But essentially, the most important thing about this plot are these numbers in the middle and the overall trends of them. So you have different phases of the drug discovery cycle, and it's a lot of money within each phase, and it takes a lot of time within each phase. And as you're going through each phase, you're reducing the number of candidates that you can potentially bring to market, all the way down to potentially just one at the end of that very long and expensive process. So what you're trying to do is to really prioritize the ones that are going to be most successful as early as possible. So you ideally want to fail fast rather than doing loads of drugs that are going to fail in phase 2 clinical trials once you've incurred all of that cost. So those principles are really, really important when you're developing drugs within drug discovery companies. And obviously, the ways that those companies go about trying to figure out which of those drugs are going to be most successful or not is they go for a series of truth-seeking behaviors. This is pulled straight from a publication produced by AstraZeneca on their famous 5R framework. And each of the R's kind of represents a really important facet within the drug discovery process that encapsulates all of these different truth-seeking behaviors. So for right target, you're looking to establish whether there's a strong link between the target and the disease, whether there's differentiated efficacy, whether there's available and predictive biomarkers. And you can kind of go through each of these and you have similar themes. So right tissue involves things like adequate bioavailability and tissue exposure, et cetera, and there's right safety, there's right patient, and there's right commercial potential. But really what you're trying to do here is collect knowledge to understand how these drugs work in as best a possible way you can. So obviously everybody here is fully aware that there's lots of data out there in the literature. That's what you've all spent your careers trying to do, capture this and model it and structure it. And we know that this is very, very difficult. We know that there are ambiguities out there for things like gene names and all of the different ways that they're described in context and in relationships with other drugs. This data is really, really hard to capture accurately. And so the kind of best way that we've gone about it for many years is to do it manually. We've had people manually putting this data into databases. But as a result, we're often missing a lot of the data that's out there sat within text. We've either not gone out there to get it directly, or we haven't yet had the time to go out there and curate it. So, there's lots of different ways you can measure this, but something like 80% of healthcare data could be inaccessible to the kinds of analyses that drug discovery companies are typically looking to do to understand how their drugs work. So at BioRelate, we focused on trying to extract some of this data using NLP, and particularly focusing on the more difficult data that you typically have human beings read through and curate. So we've built a platform that aims to achieve those kind of things. It's called Galactic AI. And in essence, it takes in lots of different sources of text, things like journal articles, patents, clinical trials, those sources of data that might contain useful things that help you answer those questions we saw in those earlier slides. And so once we've ingested all of that content, we've then got software that's aiming to capture and structure that data, and particularly the relationships. So that's really what the company has focused on over the years, trying to accurately capture relationships like cause and effect, like service expression, like biomarker relationships, which are documented within those particular pieces of research. And so at the result, as a result of trying to curate that data, we have this ginormous data set at the end of it, which we then give access to our customers via different entry points, by web applications, APIs, or even the data set itself. So that's what our platform does. And how it works with generative AI, which I think is an important theme these days, is we're using some of the generative AI itself within the platform. So we're big fans of transformers. We have been for years, and now that there's large language models, it's even more interesting, and you can use them in certain use cases, particularly once you fine-tune them to really improve the structuring of that data to produce the actual data set itself. But then once you have the data set itself, that's a very powerful resource in combination with those generative AI APIs to do RAG-like strategies. We like to use the term knowledge anchor to really help reduce things like hallucinations and get the traceability that you're looking for that typically is lacking from most out-of-the-box generative AI tools. So that's how we like to use generative AI at BioRelate. But what I'm going to do today is I'm going to try and show you some examples of what you can do with some of the data that we capture from that text that hopefully give you some inspiration for all of your curation-like activities that you might be involved in yourselves. So we're going to imagine here that we're starting a new company, but we don't really know very much about biotech companies or even something as broad as oncology. So let's start with a very generic question, such as what are the most promising up-and-coming targets in clinical studies and research for oncology? So if we went out there and tried to answer this question in the most basic way possible, we could literally just ask, how many times are protein names mentioned within oncology research articles or oncology clinical articles across 2023? And these are the results that you get back. And for people here that have studied biology and been involved in drug discovery research, these aren't really that interesting. They're really boring, in fact. These results are the proteins that you get in essentially every single area of biomedical research when you're trying to find drug targets, because they're very common and generally speaking, they're not going to make good drug targets to start focusing a new biotech company on. So this isn't a good way to try and find new and up and coming drug targets. So we might take a different approach, and we might look to see what proteins could be trending within clinical oncology research, let's say over the last couple of years. And so what you can see here in this plot are the proteins that have continuously increased in terms of publication rate over the last couple of years, just taking a very basic linear regression approach to calculate that rate of increase. And you get some slightly more interesting proteins here. You get TYMS and SHBG, but still, we're not really kind of pinpointing the ones that are most interesting. You've got STAT3, and these ones are being mentioned in clinical oncology research, i.e. they've been out there in a clinical trial somewhere. So people probably know about these proteins already if you're an oncology researcher. So perhaps these aren't the best ones to go after if you're trying to start a biotech company either. So this might be a slightly better approach to trying to find the interesting proteins to look at. Here we got ones that haven't yet featured within oncology clinical trials, but they're emerging within other research articles. So you can see in the last couple of years, particularly, there's been a dramatic increase in the rate of publications for each of these proteins, and they've never been published within a clinical trial before. So these are potentially interesting candidates to start looking at. So if we take one of those, the first one on that list, this is IFI6, and you can see that the kind of, you got the increase in publications over the last few years for it. And if you look at some of the titles from those articles, it looks like an interesting protein for oncology research. It's talking about how it's potentially involved in breast cancer cells, how it's involved in colorectal cancer. So we started to get some hints that this protein might be an interesting protein if you're looking to start a new biotech company in oncology research. But we haven't really got very much detail, and we certainly haven't got to even start to think about answering some of those more complicated questions that you need to ask within drug discovery. So this is where we can start to look at things like cause and effect to help us better understand these targets in more detail, particularly around the linkage to diseases and their potential efficacy. So first of all, quick explainer. What's the difference between something that's causal versus correlative? And I'm a big fan of memes, and this is one of my favorite memes. It shows decreasing numbers of pirates against increasing global average temperatures. And this is very obviously a correlation. There's absolutely no way in hell that the number of pirates are influencing global average temperatures. And this is because correlation is typically, or it is a statistical relationship, and it does not imply causation. And we spend lots and lots of time trying to differentiate the two. But general rules of thumb are that causes can be proximate, or they can be closest to, or they can be ultimate, the real reason. So this might be akin to the person that fired the gun versus the country that legalized the firearm. And another really important difference between cause and effect and correlation is that cause and effect is directional. That's really, really key. And causes must occur in the past if it's a directional event. But I think probably the most important thing is that causes usually have rational explanations. That's really, really key too, which obviously, again, looking at this plot, you struggle to find a rational explanation for how this particular correlation could be a causation. So if we try and apply this logic to mining relationships within text and looking for things like cause and effect, what we're typically looking for in systems biology are for changes in abundance or activity. So I've got a very, very simplified statement here taken from a piece of research that's talking about our protein again, IFI6, augmenting HSF1 activity. And so we all know that if we want to accurately capture those entities, our first step in the process is to normalize them to an ID. So I don't have to educate you all on what that process involves, but we know it's challenging, and we know that there's all the issues around precision and recall doing those particular activities. But then if we're looking to establish that there's a relationship, we need to figure out which is the cause and which is the effect. And this is a very easy example. We can see that IFI6 is the thing that's potentially having the impact on HSF1. And what we're also looking to do, particularly if we're looking to model this data in a property graph model, and when we're putting it into a knowledge graph, is to understand what the overall relationship type is, whether it's having an increased effect on that particular protein or a decreased effect on it. And you can see this is an increase, and it's doing it via augmenting it, which is the verb we might want to capture as a property in this example. So once you start to model data in this way, it then means you can start to aggregate data in this way, which then starts to reveal some early kind of insights into what's going on within the world of IFI6. So what you're looking at here are examples of the relationships that have been found between IFI6 and various different diseases. And you can see I've highlighted one here, the relationship between IFI6 and breast cancer. And you can see it's talking about how it's promoting its metastasis, it's inducing mitochondrial redox reduction, which facilitates breast cancer metastasis, et cetera, et cetera. So we're getting some early insights here that IFI6 could be a potential target for breast cancer. But still, we're not really understanding the mechanism behind how this particular protein is affecting this disease. So going back to our cause and effect examples, and you saw the one previously about IFI6 augmenting HSF1 activity, we can now start to begin to stitch these relationships together to try to build hypotheses as to how that particular protein might be affecting other things downstream of it. So we might have a separate piece of research that comes along after the piece of research that was talking about IFI1 affecting HS1 activity. this one might be saying HSF1 mediated the upregulation of LDHA. So a completely different publication published 11 years ago in an entirely different piece of research. And then we might have a third publication that comes along talking about how LDHA silencing suppresses breast cancer tumorigenesis. So we've got the beginnings of a hypothesis now. At least we've got one path that may begin to explain how IFI6 affects breast cancer. But there's a lot more that we can do with this to try and understand exactly what might be going on here between this particular target and this disease. So the next step we can do to kind of take this up a gear is we can then aggregate all of those different paths and push up the ones that are more statistically significant to give us a much better understanding of how IFI6 works across more of a systems-like representation. And what you can see here is a Sankey plot, and you can see that path from the previous slide represented with it. Maybe it's not so clear on these screens, but you can also see all of the others as well. And these are being ranked by specificity, which helps reduce that study bias and helps bring up the ones which are more likely to be the explanation for how IFI6 could be affecting breast cancer. And we can even do things like measure the overall significance of this particular target against this disease, which is kind of interesting now. So we've gone from some early research talking about IFI6 and breast cancer to now potential explanation for how this particular protein affects it. But how useful is a plot on its own if you're a drug discovery company and you're then trying to practically put this into reports and everything else? It's probably, you're probably going to want to go into each of those links and still read each one of those pieces of research to try to figure out exactly how this particular mechanism is working. So this is where our friend generative AI can come in and help us to provide that level of explanation. So what this is doing now is it's taking each link behind this plot and it's summarizing it to give us the overall mechanism of how IFI6 may be affecting breast cancer. And I won't ask you all to read this because it's probably quite far away for some people, but there are some highlights here that say things like it's complicated the understanding of IFI6's overall impact on breast cancer. So there's some contradictions within this data, and it's basically saying that we're not so sure if this is a great target or not for breast cancer. So that's kind of useful, particularly if you can get to this point very, very quickly, and you can spend far less time trying to investigate all of this data, either through reading these pieces of research or even doing experiments within the lab to try to corroborate them. So that was obviously one example from taking something that was known. So people were talking about IFI6 and its potential impact against breast cancer already. But now let's suppose we want to go out there and look for something that is yet to be talked about as a target for breast cancer, and whether we can use the data in that way. So can we find novel target disease links? So how we might think about this is really what are the levels of knowledge, what's the strength of knowledge that we can use to understand how novel something is. So I like to think of cause and effect, or things that are like cause and effect, relationships between things, as being quite strong knowledge in the spectrum of data that's out there within the literature. And then we can move down to things like co-mentions within sentences and co-mentions within documents. And the kind of linkage between any two things within co-mentions starts to degrade, particularly as you get into a kind of document co-mention type setting. But then when you get to a publication where two things have yet to be published together at all, so a protein and a disease has not been mentioned in any particular research, then we can consider that entirely novel, at least novel from the articles that we've ingested and we've been able to extract the data from. So we can use this trick again of connecting together cause and effect relationships to try to expose the novelty, to try to link together these proteins and diseases that have yet to be mentioned within pieces of research before. So you may have a cause and effect link described within one piece of research between A and B, and then another piece of research between B and C, and then C and D. And obviously, you can link A to C, and you can link A to D by hopping across these different relationships. So this is a multi-hop like approach, you might say. So if you do that, and then you start hunting for proteins and diseases that haven't been mentioned together before, but do have a number of different connections that link them, then you can potentially start to find some interesting novel candidates that have not yet been studied out there within the literature. So here is an example. This is TRIM64. It has 11 links between it at depth 2 and then 2000 at depth 3. And if you do an enrichment test at depth 2, you get a significant result using the hypergeometric mean. So this is potentially an interesting candidate. Again, if you start to use these Sankey plots and generative AI, you can start to see whether this is something that you want to look into in more detail. So now we've taken all of that knowledge from all of these different pieces of research to generate a mechanistic summary as to how TRIM64 impacts breast cancer, despite the fact nobody has yet to publish a piece of research talking about TRIM64 and its relationship against breast cancer. So it's kind of interesting. And again, if you read this one here, it actually turns out that it seems quite complicated and that it might not be the best target after all. So there's kind of one more gear you can go up here, which is rather than looking just at breast cancer, can you look at this systematically across all diseases and all potential targets? So if you were to take this from a very simplistic approach, you can see that there are about 721 possible single protein to disease pairs if you put them on a matrix. And this is a very simplistic matrix. This is literally just single protein, human protein coding genes against 40-odd thousand well-known diseases and phenotypes. And it's ignoring all of the different flavors of targets that you get, things like phosphoproteins, miRNAs, chemicals, etc. Yeah, all of the kind of potential targets that might not necessarily align with being single human proteins. So yeah, still quite a big number though, 721 million. And if we align those pairs against the different strengths of knowledge that I was trying to show you earlier, this is how many of those pairs are represented across research. So you can see at a document co-mention level, you've got about 35 million pairs of proteins and diseases mentioned. And when you get down to cause and effect, you've got about 714,000. So only 5% have even been published within papers together. So 95% of the space is dark, which is kind of interesting. So again, if we use our trick of connecting together cause and effect relationships across research, we can increase that 714,000 unique pairs upwards. So we get to 12 million at depth 2 and we get to 89 million at depth 3. And obviously you can run significance tests again to reduce these numbers down and throw out the ones that are probably there just by chance. So here's one that's really, really interesting from that big list of potential proteins. So this is RBMS1 and against cardiomyocyte hypertrophy. It's got 72 links at depth 2 and 783 at depth 3, but it's not been mentioned together as a pair in a piece of research. So that's really, really significant. And if we look at the plot and we read the summary that we get from generative AI, it's an extremely compelling drug target. So we literally know nothing about cardiomyocyte hypertrophy or this protein, and now we've suddenly got a really interesting target to go off there and do some research on. And obviously, you could focus this down into any particular area you're potentially interested in, and do all the other types of question and answering things that you want to do with this data. So I think this is amazing. This is really, really exciting. And this is the power of data once you have it and once you've captured it and structured it through doing all of this lovely ontology work that you all do. So yeah, thank you very much, and I'll take some questions.
Very nice work. What I see in the last slides is that a lot of genes are transcription factors.
Mm-hmm.
Have you taken into account the gene function? Because if you do that, you probably would even have more, let's say, relevancy, because now you have an overrepresentation of genes which are connected by nature to a lot of other genes.
Yeah. So really, really important question. So this is probably the most generic possible way to analyze the data, right? So if you were going to do this well, you would filter out certain data types that you weren't interested in. You would potentially reduce it down to certain cell types, an area of context that you would be interested in. And you would get to a point where you find those targets that really fit the profile that you're looking for. I've literally just done this through taking the most basic possible approach to illustrate that things can be connected together across research in that way, and that with things like generative AI, you can use that to potentially corroborate what's interesting and what's not. But typically, when people are doing this, they're taking the data and they're doing exactly the kind of things you're talking about here, which are really, really key. I should also point out that I'm not a drug discovery scientist. I'm just a data enthusiast, like lots of you here today.
Thank you for a great talk. I wanted to ask if you only use documents as a sort of information, as a source of information, or you also use ontologies, for example, gene ontology.
Yeah, great question. So we focus on mining data from text, and we're proud to say that we do it in a fair way. So our favorite acronym, and what that means is when you get to the end of having that data set, it should connect up with any other structured sources that you might have. So yeah, that then aligns with any in-house data or any data from genomics, transcriptomics, etc., that you might have. So again, you can empower these analysis further using things like tractability measures, using any other ways that you want to corroborate what is a good target versus a bad target. So yeah, what's typically missing from in-house data and from those other types of approaches is the directional cause and effect like data to help provide those types of hypotheses. So yeah, that's what our focus is, and it connects very nicely to the other data sources.
I wanted to ask, can you hear me okay?
Oh, shoot, yeah.
Here. So my question is, this very interesting piece of work, but I see sort of a different, maybe a different use case for this, and that is from the government perspective. A lot of times when you start to get into pharma, they start to talk about, like at the point where you did, we're going to pick targets. And usually where the governmental role is in all those studies that you have where they've been funded, where there's very little assurance that there will be something in the end to sell and to make money on. And so we're constantly, one of the big challenges is to justify the money that we invest in the programs that we develop. And I think there's an interest for everyone, including pharma, that government is able to justify this preliminary kind of work where we're giving grants and however mechanisms. And I think there's probably slightly different emphases in different countries. But I think universally, that's kind of government's role, is to take those very much higher risk, no promise of result kind of efforts. And this could help feed into a couple of ways. Number one is to help us understand where those connections are, because the public often, and we see this really in the COVID, was very pronounced, don't understand that the work that made the vaccines come quickly was really on the back of years of government funding. Mm-hmm. And they think, well, it was, pharma that got us this, which they did. But, I think citizens need to understand that there is a value to them for this investment. And then I also see, is it helping to, we don't want to narrow that funnel down too soon, but to help us direct our funding even in the current day. And then finally, you're essentially doing a systematic review, which as a library where I work, is something that we're very much interested in. So this was very, very fascinating, but I think, it's maybe a little tiny different path of a user for your work.
Yeah, thanks. Yeah, really nice idea. Yeah, I have to say, it's kind of hard actually, because there are lots of ways that you can apply relationship-like data into lots of different adjacencies, across the spectrum. 10 years of running a company's taught me that you need focus. So if you run off in lots of different directions trying to do it all at the same time, you're unlikely to be successful in any of those particular areas. So as we get bigger, who knows, we might go after other particular
We've
areas.
been spending a whole career trying to show the impact of work that is, it's not a widget that gets sold. It's really difficult to show the impact sometime of government investment.
Yeah, I mean, I'd love to help you. It's hard to do it all at once. But yeah, to your second point about systematic reviews, I really like systematic reviews because they are a great way to benchmark these kinds of approaches to show exactly what you can find that isn't known information, and also to help elevate the known information that's there already. But yeah, thank you very much. That was a lovely suggestion. I'll take it away and think about it.
Yeah. One more question, but let's not discriminate. So, you want me to read it? Yeah. Do you extract convention from supplementary material like tables and figures or protein interactions, often in PDF format?
Oh yeah, cool. So we're not doing table mining, and we're not doing figure mining. So there you go.
Okay.
But supplementary text, yes. And also, so the platform has been designed to process any source of text. So once you've ingested it, it gets converted into the same kind of format. So that goes for in-house documents as well. And we work with we work with all of the publishers to process their full text too. So it's not just abstracts, it's basically any text that we want. Where it doesn't work so well is when it's not really scientific text. And because obviously the models that we've trained and developed have been specifically trained and developed on scientific like text. So if we were to throw in it, random blogs, it probably wouldn't work as well. Or things like medical documents actually would be quite challenging, given that we haven't processed that kind of information before and gone after all the shorthand that doctors use. So yeah, they're the limitations of what's there. But generally speaking, it's quite a nice engineering setup.